Okay, we want to pick up where we left off, and I left a couple of things out that I need to point out with regard to putting your mind into the time when Mendel worked. At the time he was working, the general theory was that things blended. So the general theory was that I mean, they certainly saw that there were white flowered peas and they saw that there were purple flowered peas. He thought the idea at that time was when they got together, they blended into just lighter color purple peas. So they kind of blended like paint. So Mendel's theories were earth-shattering because he was bucking that particular rule. And then the other thing I need to clear up is it's only in the F2 generation that the white flowers show up. So with these true breeding plants, you're going to, if you cross the parents into the F1 generation, you're not going to have any white any white flowers. It's just when you cross the F1 generation in the F2 is when you have the white flowers. And this was all part of Mendel's scientific experiment and his observations of it. And then the other thing that I've been showing you but I haven't explained is this idea of a Punnett square. So let me go back so we can see one. This is an image of a Punnett square here. Mendel didn't use a Punnett square. The Punnett square was developed after Mendel. It was developed to support what Mendel is saying. And what you have is it's a basic way to figure out what the results could be, and I'm talking about the square that's above the Almy stock photo term you have there, and this did not come from your book, but you do have a Punnett square in your book, where you put the possible alleles of the gametes from the father and the possible alleles of the gametes from the mother, and you just bring them down and cross them. So what we have in this particular image, you have a big R and a little r, and the way we express it is the big R tends to be what we put together with dominant, so the capital letter with dominant, lowercase letter with recessive. So big R, little r, these are heterozygote alleles here, and you have then in the female you have big R, little r. You bring them down, and the first flower is homozygous dominant, so you have two big R's. The second one is heterozygote dominant. And then you have another heterozygote dominant, and then the white flower is homozygote recessive. And these are the terms that we put with this. But the thing I wanted to make sure I pointed out was this was the Punnett square. And then this is actually where we are, and what the next topic that we're bringing in, or the next idea we're bringing in, is this concept of a genotype and a phenotype. So when Mendel crossed his peas, he knew what the peas looked like, but he wasn't aware that there was a reason why the white ones were showing up in the F2 generation. So because of the effects of the dominant and recessive alleles, that white is not going to show up until you have homozygous recessive, until you have a plant with the two little r's. And so until that happens, one doesn't know what is possible to pass on to the future generations. So this genotype is what is possible to pass on, and the phenotype basically is what is expressed in that particular generation. Sometimes we say what it looks like. And so with this genotype at the top, you have two big P's. Remember, we're using the large letter, the uppercase letter for dominant. So two big P's, that's homozygous dominant. A big P, a little p, these are the possibilities from the male-female gametes that's heterozygous dominant. Big P, little p, heterozygous dominant again, and then little p, homozygous recessive. And he found that the genotype ratio is going to be 1 to 2 to 1. So 1 homozygous dominant, 2 heterozygous dominant, and 1 homozygous recessive. But the phenotype ratio is not going to be expressed that same way. You have a 3 to 1 ratio with regard to the phenotype. What these mathematics told him is that there must be it gave him this idea that there must be the recessive gene in there. Now, he did not use these terms, but certainly set him on that path in terms of the genetic makeup. And then with regard to an individual, could by looking at a particular pea plant, we wouldn't know whether they're homozygous dominant or heterozygous. And the only way that we can figure this out, until you have microscopes, etc., is to do this concept of a test cross. And a test cross is a breeding of a mystery individual, so somebody you want to know what it is, with a homozygous recessive individual. And the homozygous recessive, remember, that's going to be the white flower. We know that's going to be 2 little r's. And so if we cross that with somebody that we don't know what it is, you actually will find out, whether you find out in terms of flower color, because that homozygous recessive, the only thing that can be in the gamete is a little r. So the only thing that can come from, say, that's the if that's the female, is a recessive allele for that particular characteristic. And so we can then figure out, based on what the flower colors end up being, and it looks like I got it backward with regard to, well, maybe I didn't in terms of what you have here. But if you have a prediction, if the purple flower parent is homozygous dominant, so two big P's, then all you're going to have are purple flowers. You're going to have heterozygous, and this is in the F1 generation, heterozygous dominant genotype. And then if the sperm is if the egg is heterozygous, so big P, little p, and then you have your recessive little p, you're going to have a 50-50 ratio with regard to that. So the results will tell you what the mystery plant must have been in terms of its genotype. He also came up with this law of independent assortment. So the first one was the law of segregation. This is the law of independent assortment. I just said that the law of independent assortment in terms of gametes, that a single characteristic doesn't necessarily dictate what other characteristic will come with it. And so the law of independent assortment, which sometimes is called the law of segregation, can follow a single characteristic. And so you have, with the F1 offspring, the cross were monohybrid individuals that are heterozygous for one character. The cross between heterozygous is called a monocross. But what can happen is you can have a couple of different traits, and they can also be followed. So you can have a monohybrid cross, which would be the flower color, or you can also have a dihybrid cross. So the second law of inheritance, by following two characteristics at the same time, So a dihybrid cross, and again we're working with true breeding plants here. So they're heterozygous for both characters, and the character he's looking at here is color and whether the pea is shriveled or not. The dihybrid cross can determine two characteristics transmitted at the same time, so similarly with regard to a plant or packaged independently. And what he did find, and this is his second law, the law of independent assortment. I believe I misstated the other one. You have the law of independent assortment that each of the different characteristics will segregate independently. So just because you have a yellow flower or a yellow seed doesn't mean you're going to have a wrinkly seed also. They're all going to segregate. They're all going to be passed on independently. That it applies to different non-homologous chromosomes. There are those that are far apart. And what we're saying here with this homologous chromosomes, we're talking about, say, the pair that's chromosome 23 or the pair that's chromosome 24. If in fact the allele is found on those individual chromosomes, they will pass on together. You can think of, for example, redheads and freckles. There are certain things that we see that get passed on together, but if in fact those two alleles are, say, instead of on the 24, you have an allele on the 24 and an allele on 23, those are going to be independently assorted. Independent assortment. So the laws of segregation and assortment are both reflecting mathematic probability. But the thing of it is the mathematic probability is it's always 50-50. So if in fact you have a 50-50 chance of a particular allele being passed on to the F2 generation, and just because you have created these purple flowers, the next group of flowers can still be created purple, or still passed on the same way, because this outcome of one breeding generation doesn't necessarily predict what the next breeding generation percentage-wise can be. I don't know if I'm expressing that. The image expresses it just fine, but the idea of the way that the alleles are going to get together from the sperm and the egg is always a 50-50 chance. Then there is a multiplication rule, and that multiplication rule is basically taking those percentages or those fractions of the possibility of a particular phenotype being expressed via the genotype, and then multiplying those possibilities by however many what the dihybrid cross is expressing. So the multiplication rule, which is a concept that Mendel worked with, that the independent events will occur together, is the product of their individual probabilities. For example, if you have a 50-50 shot of a purple flower and a 50-50 shot of a purple flower with a with a long stem, then what you have is a 1/2 times 1/2 percent or possibility to have a purple flower with a long stem with regard to breeding. So what you're doing is you're taking this multiplication of probability 1/2 times 1/2 equals 1/4. And we're out of time. We'll pick this up on the next one.